to the Belong Speaker Series, Breaking Barriers. My name is Boris Worm, and I welcome you on behalf of both the Ocean Frontier Institute and also the Biology Department, both of which are located in Halifax, which is in Mi'kma'ki, the unceded territory of the Mi'kmaq people. Halifax is known to the Mi'kmaq as Chibuktu or Kipuchuk, the Great Harbor. And this is where Mi'kmaq clans held permanent villages and took advantage of the coves of the harbor since they offered protection from the elements and the constant supply of fresh water from the streams flowing down from one of the many lakes nearby. So, Nicola Smith, I hope you get to visit here sometime. There's also a wide variety of marine life in this area that provided food year-round, especially large marine mammals such as gray seals, harbor seals, and even Atlantic walruses were plentiful in the past. So today, and I think some of those changes are the topic of this seminar and how they relate to invasive species. So today we're very pleased to be sponsoring the presentation featuring Dr. Nicola Smith from the University of British Columbia with a presentation of conceptual and applied approaches to marine invasion and looking at Indo-Pacific lionfish as a model system. So welcome, Dr. Smith.
Thank you for having me.
Great to have you here. And again, today's presentation is part of the Belong Speaker Series, which has been organized to highlight the work and achievements of up-and-coming Black, Indigenous, people of color researchers in the fields of science, technology, engineering, mathematics, medicine, and health. This series is organized and led by Dalhousie University's Office of the Vice Provost for Equity Inclusion, and is also supported by the Office of the Vice President, Research and Innovation through funding provided by the Canada Research Chairs Program. So today's speaker is Dr. Nicola Smith. She is a incoming 2021 Liber Ero Postdoctoral Fellow at the University of British Columbia. She works with Dr. William Cheung, focusing on assessing threats caused by marine invasive and range-shifting species in Canada and the world's oceans under various climate change scenarios. She also aims to identify strategies to help coastal communities manage and adapt to these threats. Before that, she was a Bullitt Environmental Fellow and also a postdoctoral researcher at Simon Fraser University with Dr. Isabelle Côté, where she reconstructed the sponge fisheries of the Bahamas over the past 180 years and identified ways to make that fishery more sustainable. She obtained her Honours Bachelor of Science with high distinction from the University of Toronto in 2006, where she double majored in English and Zoology, and then in 2010, a Master's of Zoology from University of British Columbia, where she's also now, and in 2020, a Ph.D. in Biology from Simon Fraser University. And that PhD focused on the Indo-Pacific lionfish invasion of the Caribbean that has recently happened. And that's what we will hear about today. She has 10 years of experience in researching various aspects of biological invasions, coral reef ecology, and unreported tropical fisheries. So as you listen to Dr. Smith, please put any questions or comments in the Q&A sections, and we will have ample time after the talk for some discussion. And with that, I'd like to invite Dr. Smith to share her slides, and we can start this seminar.
Great, thank you. Can everyone see my slides?
Looks great.
Awesome. So good afternoon. Today I'm going to speak to you about some of my work that was done during my PhD. So we are now in the Earth's sixth mass extinction as species are being lost at a rapid rate. Major threats include overexploitation, habitat loss, climate change, and of course, invasive species, which is the focus of my work. Now, the accelerating rise in global trade and travel means that our world is more interconnected than ever before. This trend could severely impact species and ecosystems globally as it increases opportunities for species to invade regions beyond their natural range. Now, we should be concerned about invasive species like the ones shown here because they've led to native species extinctions and declines in abundances, as well as have disrupted local food webs and ecosystem services. Furthermore, invasive species are costly to the economy, as damages and control efforts associated with invasions cost national and regional economies billions of dollars annually. Now, in my work, I combine ecological theory and data synthesis with empirical fieldwork to tackle two main questions. First, why are some communities more easily invaded than others? To answer this question, I use a meta-analytic approach to examine the role of species diversity in providing resistance to invasions. And second, how can natural and anthropogenic control interventions mitigate the persistence and impact of invasive species? Here, I use the invasion of Indo-Pacific lionfish in the Caribbean as a model system to answer this question. I first evaluate the potential for native predators and competitors to mitigate the negative effects of lionfish on native prey. Then I investigate the ecological effectiveness of culling, or the physical removal of lionfish by divers, as a control intervention. As an expansion on this latter idea, I also look at how culling can subsequently alter the behavior of lionfish. Now, moving on to my first research question. In order to understand the relationship between species diversity and a community's vulnerability to invasions, we first have to understand the invasion paradox. Now, classic ecological theory predicts that species-rich environments should be less easily invaded than species-poor environments, but empirical evidence does not consistently support this prediction. While some studies report the predicted negative relationship between native species diversity, shown here on the x-axis, and invasibility, that is the community's susceptibility to invasion, shown here on the y-axis, others show a positive relationship, while others show no relationship at all between these variables. Here, I show the direction of the diversity invasibility relationship ranging from positive to negative on the y-axis. The invasion paradox refers to the fact that while most experiments yield a predicted negative association between native diversity and invasibility, most observational studies report the opposite, that is a positive relationship. Here, I have now included spatial scale ranging from fine to broad on the x-axis. Now, this conflicting pattern between experiments and observational studies is referred to as the invasion paradox, and is thought to arise because different processes control species composition at different spatial scales. Now, experiments are typically conducted at fine spatial scales where competition-driven biotic interactions such as niche complementarity are thought to be important. In contrast, observational studies are typically conducted at broad spatial scales where extrinsic factors that co-vary with diversity, such as spatial environmental heterogeneity, are thought to drive species composition. However, factors other than spatial scale have also been proposed to give rise to this conflicting pattern. Now, to better understand the potential drivers of the invasion paradox, I performed a meta-analysis in which I quantified both the strength and direction of published diversity invasibility relationships in aquatic and terrestrial systems. I used 3 databases in order to identify potentially relevant studies. This process yielded over 700 studies. After applying a series of a priori inclusion and exclusion criteria that involved considering things such as spatial scale, study type, and effect size, I ended up with 57 studies for analysis in which I was able to extract the correlation coefficient as an effect size. However, as I showed, spatial scale is thought to play a large role in the invasion paradox, but spatial scale can be broken down into at least 2 components: spatial grain, or the size of the sampling plot, and spatial extent, or how far apart sampling plots are spread or spaced. Now, I focused only on studies that were conducted at a fine spatial grain, but varied in spatial extent. Now, on this coefficient plot, points represent the effect size bounded by 95% confidence intervals, while numbers in parentheses are the sample size. Points to the right of the dashed line show a positive relationship between diversity and invasibility, while points to the left show a negative correlation. If the 95% confidence interval crosses 0, the result is considered not to be statistically significant. Here, I've shown you the overall effect size when all studies conducted at a fine spatial grain are included in the meta-analysis. However, when we divide these effect sizes by study type, as you can see here, the invasion paradox persists in studies at fine spatial grains, where experiments showed a negative diversity invasibility relationship, while observational studies showed the opposite, that is a positive association. But when I control for spatial extent, so that I have a smaller group of studies conducted both at fine spatial grain and extent, and once again separate effect sizes by study type, there's broad overlap in effect sizes between experiments and observational studies, suggesting that comparing studies with similar extents resolves the paradox at local scales. However, I uncovered systematic differences in the metric used to measure invasion susceptibility. Taking a look at this figure, we have study type on the x-axis and spatial extent and relative frequency on the y-axis. When I subdivide these studies up into these variables, I get the following mosaic plot, in which the area of each box is proportional to the relative frequency of that combination of variables. And the numbers inside parentheses once again represent the sample size. As you can see, experiments most often use some metric associated with invader performance, while observational studies frequently use species richness. Moreover, different metrics showed on average conflicting diversity and invasibility associations. So here I show the overall effect size for observational studies conducted at a fine spatial grain. When I divide these effect sizes by invasibility metric, the following pattern emerges. Observational studies that used richness as a metric showed a positive association, while those who used performance as a metric tended to show, on average, a negative correlation, although it was not significant. But I also want to point out that the sample size is quite small here. Now here I control for metric by only taking studies that use performance as a metric of invasibility. I then once again divided these studies by effect sizes, and surprisingly, I get the following results. The conflicting pattern between study types also disappears. Currently, it is not possible to disentangle the effect of spatial extent and invasion metric on the paradox, because as I have shown, both variables are systematically associated in different directions with study type. But what I can conclude is that both spatial extent and invasibility metric may drive the paradox, but more experiments and observational studies that incorporate the full range of variability are needed in order to understand this phenomenon. Now, moving on to my second question. I used the Indo-Pacific lionfish invasion of the Caribbean to ask, how can natural and anthropogenic control interventions mitigate the persistence and impact of invasive species? In terms of natural control, I wanted to know whether native groupers could act as a form of biotic resistance to invasive lionfish, either through direct consumption, through non-consumptive or fair effects, and/or through competition. I also wanted to know whether systematic culling by humans could control the size and effect and the impact of lionfish populations. Now, lionfish are native to the Indian and Pacific Oceans, where they're mid-level predators of coral reefs and feed on a variety of smaller fishes and crustaceans. Now, they were first introduced into the Western Atlantic Ocean in the mid-1980s. Now, if you watch this map, you'll see the little red dots showing lionfish spreading throughout the Atlantic. And watch particularly when 2004, when lionfish hit the Bahamas, as things really began to take off at that point. And this invasion is particularly important to me because I'm actually from the Bahamas, where we have some of the highest densities of lionfish in both the native and invaded ranges. Fast forward to 2020, and lionfish are now found as far north as North Carolina in the US and as far south as the east coast of Brazil, although the somewhat dated map only shows their distribution in 2018 off the coast of Venezuela. Now, people are concerned about the lionfish invasion because lionfish are incredibly effective predators. For instance, on some coral reefs in the Bahamas, lionfish predation has resulted in 50% declines in native prey biomass. And it seems as if they have no natural control because they have these venomous spines. So you can imagine people's surprise when this paper appeared in Coral Reefs Journal, showing that large groupers can in fact eat small lionfish. However, this was an opportunistic observation involving only 6 groupers, 3 of which had lionfish in their stomach contents. And this point takes me to my question of whether native groupers can act as a form of biotic resistance to lionfish. Now, there is currently conflicting evidence about the nature of the relationship between lionfish and native groupers in the Caribbean. For example, at the local scale of an MPA in the Bahamas, lionfish biomass is found to be negatively correlated with grouper biomass, as shown here with grouper biomass on the x-axis and lionfish biomass on the y. However, at much larger spatial scales, such as a study involving 3 ecoregions within the Caribbean, there was no correlation between lionfish and grouper biomass, as shown here once again with grouper biomass on the x-axis and lionfish biomass on the y. Now, unlike previous studies that examined the correlation between lionfish and native grouper abundances, I decided to look for the answer straight from the grouper's mouth, so to speak. To do this, I examined the stomach contents of over 200 large-bodied groupers representing 6 species that were caught by commercial and recreational fishers on 3 islands in the Bahamian archipelago. And what did I find? Not a single lionfish, thus showing that there was little support for direct predation of groupers on lionfish. But direct consumption is only half of the effect that predators can have on potential prey. There's also a large body of literature that shows strong non-consumptive or fair effects in which the mere presence of predators can cause prey to alter their behaviors so that it's more difficult for predators to detect, encounter, or capture prey. For example, cats for the most part eat cat food, but mice will still be afraid and hide when cats are around. This anti-predator behavior of mice also comes at a cost, as mice that spend more time hiding potentially have less time to forage and mate. And you can imagine that such strong effects might cascade down the food chain. Now, some species of groupers in the Caribbean are of a similar size and eat similar prey as lionfish, and can therefore potentially compete with lionfish on reefs as a form of biotic resistance. I examined the non-consumptive and competitive effects of native groupers on invasive lionfish by conducting a field experiment on coral reef patches around Eleuthera Island, the Bahamas, in which I exploited natural variability in grouper densities. I cleared all reefs of lionfish and replaced them with Thai juvenile individuals that I collected from outside the study area. Each reef therefore contained 3 to 4 lionfish in which I could identify individuals. And here you can see small bright orange marks from tagging lionfish with visible implant elastomer tags, which is convenient because you can identify different individuals by unique combinations of colors and placement of tags. Importantly, these tags glow under UV light, which was crucial for finding individuals on reefs at dawn using underwater flashlights, because we define dawn in this study as 1 hour before sunrise and 1 hour after sunrise. So you are in fact on reefs in complete darkness for the first part of the study. Now, I monitored grouper and native fish small enough to be lionfish prey abundances on reefs every other week for 1.5 months using underwater visual surveys and transects. I also recorded a series of lionfish anti-predator behaviors on reefs every 30 seconds for 30 minutes per fish, per survey at both dawn and throughout the day. Behavioral surveys took place every other week for 1.5 months. Now, there were 3 species of groupers on my reefs that eat fish to varying degrees, as shown in here in this density plot of the expected proportion of groupers for a given body size. The dashed line on the density plot of 15 centimeters total length represents the mean size of lionfish on my reefs. Predators could therefore be defined just based on species identity alone, with the expectation that primarily piscivorous species like black grouper would pose a higher predation risk to lionfish. But the other thing that's important here is the size of individuals, because fish are gape-limited predators. Therefore, based on the size of lionfish, I also defined any individual that was over 30 centimeters total length as a potential predator of lionfish, and any individual that was less than 30 centimeters as a potential competitor. In all models, I therefore compared the strength of support in the data for groupers acting either as a predator or as a competitor to lionfish based on species or size. Now, I measured 3 anti-predator behaviors by lionfish. These included hiding, inactivity, and total swimming distance. I also examined 3 factors that might explain lionfish behavior. These included the density of grouper predators defined either by species or size, the density of grouper competitors defined by size only, and the density of other lionfish on the reef, as lionfish from neighboring reefs continue to recolonize experimental reefs throughout the study. But because behavioral surveys took place at dawn, when both lionfish and groupers are likely to be hunting, And throughout the day, when both taxa are likely to be resting, I also consider time of day in my models. Now, I looked at several lionfish anti-predator behaviors like hiding, as shown here, and formulated numerous a priori hypotheses based on factors that I thought might influence lionfish behaviors. Now, these hypotheses were rewritten as mixed effects models, which I then competed against each other using AIC corrected for small sample sizes. So, for example, in this basic model, I examined the effects of 2 factors on lionfish hiding at dawn. However, these are more than just factors, as I can also imply mechanism from my models. So in this example, groupers identified based either on body size or species are acting as a potential predator of lionfish, while smaller body groupers are acting as potential competitors of lionfish. Now, one of my key results is shown here. Specifically, after competing 20 candidate models relating to lionfish hiding behavior, the top model turned out to be this very simple one, where I have grouper predator density based on size on the x-axis, and the probability that lionfish are hiding at first sighting at dawn on the y-axis. The bands represent 95% confidence intervals, while the points are jittered along the y-axis and indicate where the lionfish was observed hiding within a reef, signified by number 1, or out in the open, signified by 0. As you can see, the higher the grouper predator density on reefs, the more likely lionfish were to be hiding at dawn when they should otherwise be hunting. There was 38% support in the data for this top model, which explained 60% of variation in lionfish hiding on study reefs. Now, here is a coefficient plot of the average of 6 top models based on competing 40 alternative hypotheses related to lionfish inactivity. Points of effect sizes bounded by 95% confidence intervals. Points to the right of the dashed line mean that the factor led to an increase in lionfish inactivity, while points to the left led to a decrease in lionfish inactivity. Gray points indicate some uncertainty surrounding the direction of the effect size. Now, there's a lot going on with these results. However, the main point for our purposes here is that while the probability of being inactive increased during the daytime on reefs with increasing densities of grouper predators, the opposite was true for lionfish competitors. On average, lionfish were more likely to be inactive as conspecific densities increased. I'd also like to point out here that there is some uncertainty in my estimates around the effects of grouper predators and lionfish competitors. Moving on to lionfish swimming distance, I found that the interaction between conspecific abundance and time of day was the primary driver of lionfish total swimming distance. This model explains 65% of the variation among individuals. Here I have lionfish density on the x-axis and the natural log transformed total swimming distance of a lionfish on the y-axis. Time of day is represented by 2 colors, where dawn is in orange, while throughout the day is in blue. Once again, bands represent 95% confidence intervals. Now, lionfish swam longer distances at dawn with increasing densities of conspecifics on reefs. However, this pattern occurred only at dawn when lionfish are likely to be hunting, but not during the day. Now, this behavioral change is likely a response to intraspecific competition for food or space among lionfish, as previous studies have shown that lionfish exhibit density-dependent growth, movement, and foraging range. Finally, although I found that lionfish hid more and were less active at dawn when both lionfish and groupers should otherwise be hunting, I did not detect that this effect caused a cascade down the food chain. In other words, reefs with high abundances of groupers did not have more native prey fish due to reduced lionfish foraging. Now, there are several potential reasons for this, which I can discuss later after this talk if you'd like to. Anyway, what we can say overall is that native groupers can trigger behaviors in lionfish that can potentially reduce lionfish foraging success, but they can't fully mitigate their negative impacts on native prey. Now, taking a step back, what does this mean in the broadest sense of things? Well, large predators are in decline worldwide, causing large-scale disruptions to ecosystem structure and function. But most people only consider the consequences of the loss of consumptive effects of predators. Here, I show that the depletion of large predators can have implications that go beyond demographic effects on prey and can, in fact, also alter prey behaviors. Now, moving along to the anthropogenic means of controlling invasive species, I used a 2-year-long field experiment to evaluate the ecological effectiveness of culling, or the physical removal of lionfish by divers, on timescales in keeping with the constraints of severe resource limitation in small island states. Now, culling is commonly used in conservation as a means to eradicate non-native species or to reduce their population to levels that minimize their ecological impacts. In such instances, culling can be an effective tool. For example, culling is being used to completely remove introduced goats, cats, and pigs on islands while in North American temperate lakes, repeated removals of the introduced smallmouth bass resulted in a decrease in its abundance and a subsequent increase in the abundance of several native prey fishes. However, culling is also costly, labor-intensive, and can sometimes have unintended ecological consequences. For example, in another conservation program with the introduced smallmouth bass, culling actually resulted in an increase in bass biomass. While in Australia, culling dingoes decreased its population, but also resulted in an increase in feral cats, which then preyed heavily on native birds. Now, in my long-term experiment, the question was quite simple. How frequently should we cull lionfish in order to reduce lionfish populations locally and to halt the native prey fish decline? Now, this project took place once again on Eleuthera Island in the Bahamas, and I had 12 coral reef patches divided into 3 groups. Reefs that were culled every 6 months or biannually, reefs that were culled every 3 months or quarterly, and control reefs where no culling occurred. Now, these removal schedules are important because they coincide with the financial schedules of many NGOs and private institutions in the Bahamas. This contrasts with previous studies that show that culling lionfish can be effective, but was conducted at a monthly frequency, which cannot be financially sustained on the ground in the long term. Now, over a period of 21 months, I measured lionfish and native prey fish small enough to be lionfish prey densities and biomass at the start of the experiment, and then at 3-month intervals on both culled and control reefs until the end of the experiment. Now, a number of other factors in addition to culling frequency may affect lionfish density, such as culling efficiency, which refers to the proportion of lionfish remaining on a reef immediately after a cull, as you are not always 100% effective at removing all lionfish. Also, coral fish recruitment varies seasonally in the Caribbean, thereby leading to predictable changes in lionfish food availability. And hurricanes may also be important, as the Bahamas sits in the hurricane belt, and Category 3 Hurricane Irene passed directly over my study site 9 months into the experiment. Now, I used mixed effects models with AIC corrected for small sample sizes to test the effects of these factors in various combinations with and without interactions. After computing 17 different models, the variables that best predicted changes in lionfish density were culling frequency and the presence of hurricanes. Here I once again have a coefficient plot in which points are represented by effect sizes bounded by 95% confidence intervals. Points to the right of the dashed line signify an increase in lionfish density, while points to the left indicate a decrease in lionfish density. Now, this model explained 51% of the variation in lionfish density among reefs. Focusing only on those variables within the red box, as predicted, culling reduced lionfish densities between 60 and 80% before the hurricane. However, biannual culls were as effective as quarterly culls, as shown here by the broad overlap in confidence intervals between the treatments. Now, this result was surprising to me because one would think that culling more frequently would be more effective at reducing lionfish populations over time. But there is an explanation for this pattern. It turns out that lionfish from neighboring reefs rapidly recolonize study reefs in the system in a density-dependent fashion, such that lionfish preferentially move from high-density to low-density reefs. Culled reefs therefore act sort of like a magnet for lionfish in the surrounding areas, where reefs that are culled more frequently act like a stronger magnet. However, after the hurricane, the fish hit the ceiling, so to speak, as lionfish densities on culled reefs increased by on average a whopping 630% relative to reefs before the hurricane where no culling took place. Now, this pattern is explained by high rates of recruitment of small lionfish onto all reefs following the storm, but particularly so on reefs where culling took place. Moreover, this hurricane effect persisted until the end of the study, roughly 12 months after the storm. Now, I don't have time to talk about what happened to native prey fish with culling, but suffice to say that the culling frequencies before and after the hurricane were insufficient to halt the decline in native prey fish. So to wrap up this section, what have we learned? Well, infrequent culling offers a poor trade-off between the demonstrated conservation gains that can be achieved with frequent culling and the economy of time and money realized by infrequent culling. And secondly, stochastic events such as hurricanes can drastically limit the effectiveness of culling. Now, in an earlier study, we found another unintended consequence of repeated culling in which this activity altered lionfish behaviors such that current and future control efforts may become more difficult. Earlier in this talk, I mentioned culling efficiency, or the proportion of lionfish remaining on a reef after a cull, because you are rarely 100% effective. Well, in this study, we wanted to know why exactly it is that it is so difficult to remove all lionfish from a reef after repeating culling has occurred. To answer this question, we once again used our study system of coral reef patches on Eleuthera Island in the Bahamas, in which lionfish had been culled biannually, quarterly, or not at all at study sites over 2 years, and measured 2 lionfish behaviors. The first was alert distance, or the distance between a disturbance and when a fish first shows visible signs of alarm. In this case, the disturbance was a slowly approaching diver at 3 to 4 meters from the lionfish, while visible signs of alarm in lionfish included sudden flaring of dorsal and pectoral fins, orienting toward the diver with spine facing forward, or slowly turning and withdrawal from the diver. Now, the second behavior was the extent of lionfish concealment on reefs. For this behavior, we used a categorical scoring system from 1 to 3, in which higher numbers means that the lionfish is more hidden within the reef. So in this example, lionfish could be out in the open, as denoted by number 1, partially hidden, as denoted by number 2, or deep within the reef, as denoted by number 3. Because there was no difference in measured behaviors between reefs that were culled annually versus biannually, we pooled the results of these treatments, where we have treatment on the x-axis and alert distance on the y. As you can see, lionfish became alert at greater distances from divers on reefs where culling had repeatedly occurred than on reefs where culling never occurred. This observation could be due to 2 possible mechanisms. First, culling can act as a form of selection against bolder individuals, thereby leaving only shy individuals, which are more difficult to catch over time. Or alternatively, on reefs where culling has occurred, the individuals that are left behind may learn to become wary of divers, either due to direct experiences of a close encounter with divers or through social learning. At present, it is not possible to determine which mechanism is at work here. Furthermore, we found that lionfish tended to hide deeper within reefs at cull sites, as shown by the higher hiding score on average than on control reefs. So what does this all mean? Well, lionfish are frequently removed from reefs using pole spears, in which a diver typically needs to get pretty close to an unstartled fish for accuracy. Lionfish on coral reefs hid better and became more alert at farther distances than on lionfish on reefs where culling never occurred. This means that through time, it is likely to become increasingly more difficult to remove all lionfish from reefs where culling has occurred and not all individuals were previously removed. Hence, control efforts are likely to be less effective or require more people and/or time to achieve similar results in the future. Now, coming back to the main questions of this talk, what can I say overall? Well, development of standardized metrics is key to generate a holistic understanding of the relationship between native species diversity and invasion dynamics. Until this happens, we cannot say with much confidence why some communities are more easily invaded than others. And secondly, both natural and anthropogenic control over invaders is unlikely to stymie biological invasions at the scale currently observed for Indo-Pacific lionfish in the Caribbean. Sad but true. And with that, I'd like to thank my funding sources for this work. My former PhD supervisor at Simon Fraser University in the Earth to Oceans Research Group, Dr. Isabelle Côté. My former lab mates and members of the Earth to Ocean Research Group at SFU. And finally, I'd like to end by giving a broad overview of what I'm up to now. As announced in the intro, I'm now a Liber Ero Postdoctoral Fellow in the Changing Ocean Research Unit led by Dr. William Cheung at the Institute for Oceans and Fisheries at UBC. Here, broadly speaking, I work on interactions between marine invasive species and climate change scenarios at both global and regional scales. I'm using a variety of methods ranging from meta-analyses and species distribution modeling to risk analyses and invasion management surveys to help people better monitor, manage, or adapt to marine invasions in a changing climate. So my work centers around three main objectives. These are, firstly, to quantify and compare the impacts of introduced versus range-shifting species in marine systems globally under climate change scenarios. Secondly, to assess the risks posed by introduced species in Canada's oceans under climate change scenarios and to identify species and traits, hopefully, that pose a higher risk to Canadian biodiversity. And lastly, to identify, summarize, and assess the ecological effectiveness of current management strategies for introduced species in Canada, classified as posing a high risk to Canadian biodiversity under climate change scenarios. And this last objective will help, hopefully prompt more research into how these management strategies can be adapted under climate change, because research has shown that these management strategies are likely to become less effective under climate change scenarios. But it's hard to know what you need to change if you don't know what's in place right now. And unfortunately, with the state of the field, many people are doing many different things, but very few people have actually evaluated the ecological effectiveness of what they're doing. And with that, I'd like to end and thank you for your attention. And are there any questions?
Thank you very much for a really enlightening seminar. Everything that's possible to know about lionfish invasions and how to control them or not. Really, really interesting. I learned a lot. I've wondered about this invasion for a while. Would you mind providing us with a little bit more context as to, do we know where it came from, what the source of the invasion was, and then also what has anyone estimated the current economic or ecological consequences? And is it still spreading and gaining abundance, or has it kind of stopped in its tracks? Where is it now? And while you're answering that question, I'd like to invite everyone to put their questions into the Q&A box, and then we can go from there. Thank you.
Yeah, so lionfish were likely introduced into the Western Atlantic Ocean by the aquarium trade, actually, because they are highly popular. They're the top 10 most valuable fishes in the global aquarium trade. Like, actually, if you go on Google right now, you can still find fish, a single individual could go for up to $100, even though it's invasive, you can still buy them online. And so it's thought that over time, individual pet owners gradually released the lionfish into the ocean, either because the lionfish became too big for its tank, because it can grow up to about 40 centimeters total length, or the lionfish basically began to eat all the other fish in the tank. And so you no longer wanted that lionfish, but you didn't want to kill your pet Fred. So instead you released him into the ocean. And over time, many people doing this, and the fish became invasive. Now, in terms of what impacts are they having? So it varies in the northern, right? Currently, what we know with the state of things is the impact varies in the Northern Caribbean versus in the Southern Caribbean. And we're not sure if that's a factor due to the time of the invasion or if there's something else going on there. But in the Northern Caribbean, we've seen very strong impacts where lionfish resulted in native prey fish declines of up to 65% on natural reefs. But also we found in places like Belize, they're eating native endemic species. And they've also found that they're eating a species that was actually completely new to science. Like, no one knew this fish existed until they did lionfish stomach contents, and they found this fish, and they're like, wow, so they got to identify a new species. So you can possibly lose something without even knowing that it's gone with this invasion. Whereas in the Southern Caribbean, people are finding little impacts or little to no impacts of lionfish. So there's kind of this conflict going on in the literature in terms of what indeed is the impact of lionfish. And that's kind of the same idea we found in terms of the relationship between groupers and lionfish on reefs with the abundances. Depending on the spatial scale you look at the location, the results seem to vary. And in terms of, is this invasion still going? It is. It's still got a lot of steam in it. It's now in Brazil and spreading. And it's predicted that it's going to spread straight down to, I keep mixing up Paraguay and Uruguay, whichever one is on the coastline. It's basically going to spread to that point, and it's only going to stop there and be stopped by winter bottom temperatures. And actually, it's northern range in North Carolina in the US. It stops there because of winter bottom temperatures. However, you can find them in the summer straight up to Long Island, New York. And that's the juveniles go up there in the summer because of the Gulf Stream allows the waters to be warm enough. But in the winter, they all die out because the water temperatures become too cold. So we say that their northern limit is North Carolina simply because that's as far as they can have established populations, but they can be found beyond that range. And I forgot, was there anything else to your question?
No, that was essentially, I just wanted a little bit of context on what's happening now and what are the costs of the invasion. Oh,
another cool thing about the invasion, I forgot, well, not cool, but lionfish are now invading the Eastern Mediterranean as well. And they've entered through the Suez Canal. And so we have this invasion occurring now in the Western as well as the Eastern Atlantic. And at least the researchers in the Mediterranean think it has a bit to do with climate change, making the environment a little bit more hospitable to lionfish right now. So it's becoming a really big problem in the Eastern Mediterranean as well. And there are technically 2 species of lionfish invading the Atlantic, whereas only 1 species is currently invading the Mediterranean.
Mm-hmm. Mm-hmm. I did not know. When did the Mediterranean invasion occur?
I think they started to see in the mid-2000s, around 2011 or so, they started to see them showing up. And now they're like spreading along the Mediterranean.
Oh. So we have a couple of questions in the Q&A section now. Maybe I'll just read them out. So Lorena asks, thanks, Nicola. I really enjoyed this. I'm quite interested in your upcoming work looking at Canadian invasions. What are some of the worst invasion species in Canadian marine waters right now, and what sorts of efforts exist now to control them? Are these effective? If nothing too bad, what species are we predicted to get in the future? So current and possible future invasions in this country.
Well, I've just started this postdoc in September, so I'm still trying to get up to speed on Canadian biodiversity. But what I do know so far is that you have the European green crab, which is invasive throughout the world. It's a pretty nasty invader, and it outcompetes native crabs as well as it destroys eelgrass meadows, which serves as habitat for a lot of native fishes and so forth. It's actually in British Columbia right now as we speak. And so people are currently doing models to predict where it's going to show up next. And in terms of control efforts, I'm not too sure exactly what they're doing. They may be doing something. I'm just not up to speed on the literature as yet. In terms of future invaders, I can't answer that question as yet, but I will be able to, once I finish my species distribution models, I'll be able to let you know what's coming and what should we be looking out for the most. And hopefully not just species, but maybe some traits of certain species traits that are more likely to pose higher risk to Canadian biodiversity. Because this is going to occur in 3 oceans, both the Arctic, the Pacific, and the Atlantic. So it's going to be a pretty big study. But I'm like I said, I just started in September and I'm still figuring out the logistics of like, where are my office keys, sort of thing.
All right, yeah, we've all been there, moving places. I know that lobster fishermen here tend to be concerned about black sea bass moving up the coast, and they're a predator of juvenile lobster. I actually have another question on predation, but there's a follow-up question now from Lorraine. What types of management measures, if any, are in place to avoid the provinces related to these species? Or if you can't answer that yet, what kind of management measures do you think work, given your experience with the lionfish model? It appears that what people have tried was not very effective in the long run. Is that true? What is your recommendation?
Well, so in terms of the lionfish invasion, it's kind of complex because, well, we found that culling doesn't really work long term, but it's a good sort of public outreach sort of activity to say, hey, there's an invasive species, let's do something about it. But so you could decrease lionfish numbers, but like my studies show, you actually don't halt the decline in native prey fish, which means you're not minimizing the biodiversity effect. However, if you cull at really high frequencies on reefs of concern, for instance, which is happening in marine protected areas, as well as by the recreational dive industry on some of their sites, you can be effective. But in terms of broader regional scale control methods, what has been put in place is two things. One is a commercial fishery for lionfish, because they're actually quite tasty. They taste like groupers. And once you remove those venomous spines, you can actually eat the flesh. And they're considered to be a native delicacy in their native range. And that has mixed results in the sense that it's great because if you want to get rid of something, the best way to do it is fish it out. However, what's happening is that lionfish, they have very strong site fidelity. So if you see a lionfish on a coral reef on a certain rock, you can come back 6 months later and chances are it's going to be that same individual on that same rock. And the problem with that is that fishermen also know this. And so when they see a lionfish and it's quite small, they don't shoot it because they get little to no money for a small individual. But if they come back 6 months, 8 months later, when that lionfish is much larger, suddenly that same individual can double, triple in price. But at the same time, it's sitting on that reef, eating a whole bunch of other species and reproducing because it reproduces once every 4 days in the Atlantic. So and it becomes reproductively active, like within the first, I think within the first year. So it can do a lot of damage pretty quickly. So there's also the problem that hasn't happened as yet, but what they're concerned about is that once people develop an economic dependency on a certain species, the incentive to kind of wipe it out kind of disappears. And that's what's happening in Honduras, where they have an entire factory for export solely just on lionfish, because you can export them to upstate New York and Whole Foods sells them. So it's quite lucrative. But if you start to create a whole market based on the single species, the conservation message might get lost in there. And thirdly, we're kind of concerned about people basically mimicking. If you see the success of the lionfish in terms as a marketable fish, a high price fish in niche markets in North America, you're afraid that people might start introducing them to other places in the world to basically get a foothold into those markets. So you can have copycats intentionally introducing lionfish. So with a commercial fishery, it seems to be the most effective in terms of, get a lot of fish over a large range, but there are a lot of caveats that need to happen with that, and a lot of public outreach and education needs to go along with those efforts. And in terms of the second part of your question, in terms of what sort of techniques are used in Canada and elsewhere. So culling, like I said before, in part of the talk, is quite effective, but it depends on what time of the invasion, what state of the invasion you get it in. And it usually doesn't work too much in marine systems, unfortunately, because of the wide dispersal capabilities. There's also, what you can do is you can release sterile males into the environment. And that works at least with insects. I don't know if anyone has tried that in the marine system as yet. There is, let's see, there's biological control, which of course can always be like biocontrol, which can also be kind of risky. That's when you kind of release a native predator, competitor, or parasite or something from the native range, and you kind of introduce it into the invaded range to kind of control the invasive species. And of course, that didn't work out so well in Australia, so people try to stay away from that. And then, of course, what we have is natural biotic resistance, which is what I talk about in my talk, which is trying to basically buffer up native predators and just diversity in general, which seems to in some cases seems to be able to resist invasions, if not resist them, at least minimize the impact that it would have otherwise in species poor environments where they're little predators and this sort of thing. So yeah, depending on what stage you get it in, the different stages of the invasion process require very different management strategies. And so if you're, yeah, so it depends on the stage and it depends on the system in which you're in. What works and what doesn't work is still up in the air. Unfortunately, invasion biology is still a field where everything is very context specific, and we're trying to move towards generalities, but it's very difficult to do in invasions.
I was particularly struck by your time-lapse video of the invasion, and that there were a few years where it might have been possible to constrain it due to local occurrence, and then just burned out of control very, very rapidly, right? I thought that was a great education on when to fight an invasion, right?
Well, that's the thing with most invasions. There are a lag period where you have a species and it sits around in the environment for a while and it seems not to be doing anything. Either it's so low abundances it's not detected, or you detect it, but it doesn't seem to be doing much and there don't seem to be many, so you don't allocate resources towards removing that species. And then something happens, it hits some sort of critical threshold and then it just kind of takes off.
Yeah, we have 5 minutes left. I really don't want to dominate this discussion period with my own curiosity, although it is there. So if anyone else feels compelled to ask a question, please do that now. While people are thinking, just a quick question on predation. So I was interested in the group. I mean, the grouper did eat lionfish, at least in one location, although there was no broader correlation. Do the lionfish eat grouper, and could that result in a multiple stable state scenario where either grouper, when they're abundant as adults, control lionfish, presumably as juveniles, or when groupers are depleted, say by fishing, lionfish as adults can control groupers as juveniles and such prevent their predators from reaching a size where they could become a threat?
Well, there has been some evidence of adult lionfish preying on grouper juveniles, but it's been very rare. So we're not sure about the frequency in which that event happens. But it's definitely possible because lionfish will basically consume anything that can fit in its mouth when it's an adult. And I think I saw a question in the chat about predators and competitors to lionfish in their native range. Well, that's the funny thing, in their native range, no one knows what controls lionfish abundances there. There are about 14 different species of lionfish in the native range of the Indo-Pacific. And so they think it might have to do something with, you know, intraspecific competition might be playing a role there. But in terms of predators, there's only one example of Pacific connet fish, a really big individual with a juvenile lionfish in its stomach, and that was way back in the 1980s, and that was just one observation. Since then, though, they have found that moray eels, both in the Atlantic as well as in the Indo-Pacific, will feed on lionfish. So that's a potential predator, and sharks also will occasionally eat lionfish with no ill effect. And so people have this really bad idea that's going on in the Caribbean, which is just so quote unquote train sharks to eat lionfish, where they spear lionfish and feed them to sharks. And of course, we try to warn against that because you basically make sharks form this association with people and food, which is never a good association to make. And just because a shark eats a lionfish that is injured doesn't mean it's going to go after a lionfish that's completely healthy. Any fish will go after something that is injured. Doesn't mean it's going to go after a big healthy lionfish with lots of spines on its own. So the moray eels look like they're the best hope right now. And with groupers, even if groupers were an effective predator, the problem is they're so heavily fished throughout the Caribbean that their numbers just aren't big enough to control lionfish anyway.
Interesting. And thanks to Kevin for that question, which was in the chat. Other questions, either in the chat or the Q&A? Anyone? If not, we're almost at the top of the hour. So Nicola, really like to thank you very, very much for making time early this morning, your time, to talk to us and talk about your thesis research. Wishing you all the best with your postdoctoral program. And hopefully once you're wrapping that up, we can hear from you again on what came out of your research at UBC.
I would love to. Thank you so much for having me.
Great to see you, and thanks everyone for joining. And just a quick announcement that we have another seminar in the Belong series on a similar subject of marine invasions by Alexander Davis, coincidentally also Liber Ero Postdoctoral Fellow at the University of Alberta. And that will be in an hour at 3 p.m. our time. If you haven't received an invitation for this, check the website of the Belong series, or just send me an email at bworm@dow.ca and I can send you the link. Thanks again to everyone, and especially to our speaker today, and have a lovely day.